Hello everyone and welcome to today's session. So today we'll be discussing the mock test, okay? And we'll also discuss some practice questions related to these topics. So let's begin with this first question. What is the first question? Which type of mutation causes a frame shift in DNA reading? Okay. So what happens due to mutations? The DNA sequence changes and the reading frame changes. Okay, what do you mean by reading frame changes? Okay, so the codons are read in a certain way. So due to this mutation, the codon changes. Clear? So now the new codon codes for some another amino acid. You have these four options. First option is substitution of a base pair. Second is deletion of 3 consecutive base pair. Fourth is insertion of 1 base pair. And fifth is duplication of a chromosome segment. What will be the correct answer? Correct answer here will be option number C, insertion of 1 base pair. So see, frame in what happens in frame shift mutation, there is an insertion or deletion of 1 base pair. Okay, and because of this, there is a shift in the reading frame. Clear? So there can be insertion of 1 base pair or deletion also of a base pair. Okay, suppose there is addition of adenine or there is a deletion of some base. So this will cause a change in the reading frame. Now next question is the woman carrying a gene for color blindness. The woman is a carrier here. Okay, she is unaffected but she is carrier. She is marrying a normal visioned man. What is the probability that their son will be colorblind? So remember, colorblind is a X-linked recessive disorder. Okay, means it is linked to the X chromosome, and it is recessive means the mutation, this color blindness, this is a recessive. It will only appear in homozygous condition. Okay, so what will be the answer? 0%, 25%, 50% or 100%? See, the answer here will be option number C, that is 50%. So you can see man is normal, father is normal. Okay, and father, they have X and Y chromosome. Now the male can only transmit Y chromosome to the sons. Clear? And X chromosome, it will transfer it to the daughters. But the man is normal, so it will transfer only, or it will transmit normal X chromosome to the daughters. Now female is a carrier, means females have X and X chromosome, they have a pair of X chromosome. Now this female, which is a carrier, she can either transmit X chromosome, which is having the color blindness gene, or she can also transmit X chromosome having the normal gene. That's why there is a 50% chance of the daughter, of the son being colorblind. Okay, clear? So what you can do, you can make the gametes and then you can make a Punnett square. Okay, so the probability of the son being colorblind will be 50%. One of the son will be normal, another son will be colorblind. Because in males, what happen? Even if one mutated gene is present on the X chromosome, the disease will be, you can say, observed. Because the normal you can say gene is not present on the Y chromosome. Next, in phenylketonuria, the affected individuals lack the enzyme required to convert. So phenylketonuria, it is a metabolic disorder, and here a gene is mutated, and due to mutation in this gene, an enzyme is not synthesized. The name of the enzyme is phenylalanine hydroxylase, and this enzyme converts phenylalanine to what? Okay, or does it convert tyrosine to phenylalanine? Does it convert phenylalanine to tyrosine? Okay, or it converts valine to glutamic acid or glutamic acid to valine. So you have to tell the function of this enzyme. What will be the correct answer? Okay, so here the correct answer will be option number B, means phenylketonuria to tyrosine. So this enzyme, phenylalanine hydroxylase, this convert phenylalanine to tyrosine. These are both amino acids. When this enzyme is not being produced, then phenylalanine will accumulate in the body. Phenylalanine then gets converted into phenyl pyruvic acid and other harmful compounds. And these compounds, they can cause mental retardation. And they can also cause some other effects like reduction in skin pigmentation and hair pigmentation as well. Remember, this is a very good example of pleiotropy also, where one gene affects multiple traits. Now next, which chromosome disorder is characterized by karyotype 47XXY? So you should remember the karyotype of all the chromosomal disorders. The chromosomal disorders are caused due to change in the number of chromosomes or the structure of the chromosome. Okay, now you can see they are saying 47 means there is one additional there is one additional chromosome. This additional chromosome can be one additional autosome or one additional sex chromosome. Here you can see there is one additional X chromosome here. So this karyotype is related to which chromosomal disorder? What are the options? Turner syndrome, Down syndrome, Klinefelter syndrome, or option number D is phenylketonuria. What will the correct answer? The answer here will be Klinefelter syndrome, option number C. So Klinefelter syndrome is observed in males that have one additional X chromosome. Okay, so males, they have XY. Here the males have one additional X chromosome. So these chromosome disorders, they are usually because of non-dissection of the chromosome. And here the person will be having one extra chromosome here. So in total, there will be 47 chromosomes instead of 46. Next question, which of the following is a Mendelian disorders? So genetic disorders, they are of two types, Mendelian disorder and chromosomal disorders. Mendelian disorders are due to mutation in a single gene. Which is a Mendelian disorder? Turner syndrome, thalassemia, Down syndrome, or Klinefelter syndrome? What is the correct answer? Here option number B, thalassemia, is the correct answer. So thalassemia is a quantitative disorder where less amount of hemoglobin is produced. So basically, less amount of beta globin chain is produced. Okay, so this is due to mutation in genes such as HBA1 and HBA2 or HBB, depending on the type of thalassemia. So there are two types of thalassemia, alpha thalassemia and beta thalassemia. In alpha thalassemia, alpha globin chains are affected. In beta thalassemia, there is a reduced synthesis of beta globin chains. Okay. Now, next one, chromosomal aberrations are commonly observed in gametes, cancer cells, stem cells, or neurons, also known as the nerve cells. So, what would be the correct answer here? So option number B, cancer cell is the correct answer here. So, cancer cell, they keep on dividing. Okay, they have lost the ability, you can say, of contact inhibition. They keep on dividing. They divide very rapidly, and during cell division, there can be a lot of chromosomal aberrations. Okay. Next, which of the following is an autosomal recessive disorder? Okay, so they are saying autosomal means it is linked to autosomes. It is recessive disorder means the mutation which is in the genes present on the autosomes. This mutation is recessive. Okay, and it will only be expressed in homozygous condition. Which of the following disorder is autosomal recessive? First is hemophilia, second is Down syndrome, third is sickle cell anemia, fourth is Klinefelter syndrome. What would be the correct answer? Option number C is sickle cell anemia is the correct answer. So what sickle cell anemia is due to a point mutation. Okay, what is point mutation? There is a change in the single base pair of the DNA. So in sickle cell anemia, there is a change in the single base pair at the 6th position of the beta globin chain. Clear? So this leads to amino acid substitution. Instead of glutamic acid in the normal hemoglobin gene, okay, this glutamic acid replaced by valine at the 6th position of the beta globin chain. This causes hemoglobin polymerization and the shape of the RBC changes from biconcave to sickle shaped. Next is, which mutation type involves the loss of a DNA segment? Okay, in which type of mutation the DNA segment is completely lost? Options are insertion, duplication, deletion, or inversion. What will be the correct answer here? The answer will be option number C, deletion. Deletion causes loss of the DNA segments. So genes are completely lost. Okay, because a DNA segment is being lost. Next is, which of the following is a result of point mutation? The point mutation we've already discussed. It is a change in the single base pair of the DNA. Which disorder is due to a point mutation? Down syndrome, sickle cell anemia, Turner syndrome, or Klinefelter syndrome? So this is a very straightforward question. What will be the answer? The answer will be option number B, sickle cell anemia. Sickle cell anemia is due to point mutation. Down syndrome, this is known as trisomy of the 21st chromosome. Here there are 3 copies of the 21st chromosome. Then you have the Turner syndrome. Turner syndrome, this occurs in females, and these females lack one X chromosome. Infector syndrome, this occurs in males which have an additional X chromosome. So all of the other, except sickle cell anemia, all of the other are chromosomal disorders. Now, UV radiation is an example of carcinogen, mutagen, teratogen, or oncogene. So, carcinogen, this can cause cancer. Mutagens, they cause mutations. Teratogen, these are the agents which can cause birth abnormalities. So, they cause abnormalities during fetal development. And then the last is oncogenes, genes which can cause cancer. So, what will be the correct answer here? The correct answer here will be option B, mutagen. So it is a physical mutagen. It causes mutations. And due to mutations, there is a change in the DNA sequence. And due to change in the DNA sequence, the protein which this, you can say gene codes, that protein changes. And if the protein changes, that the phenotype or the trait will also change. Or there can be some another phenotype. Next, according to Mendel's dihabit cross result, what is the phenotypic ratio observed in F2 generation? So they are talking about dihabit crosses. What are dihabit crosses? Crosses between individuals which differ in 2 traits. And these 2 traits are controlled by 2 different genes. So what is the phenotypic ratio in F2 generation? So your options are 3:1, 9:3, 3:1, 1:2, 1, and 1:1, 1:1. What will be the correct answer? The correct answer here will be option number B. When the genes they are sort independently, then you get the phenotypic ratio of 9:3, 3:1, means you are getting 4 phenotypes here. And in the F2 generation, the recessive character, it reappears again. Okay. The next, which statement about the XY sex determination system is correct? Okay, so they are talking about XX and XY sex determination system here, which is observed in humans as well. So where the females are heterogametic, means are the females are producing 2 kinds of gametes. Then B is males have 2 different sex chromosomes. Okay. Third option is autosomes differ between males and females. So do the number of autosomes, they differ in male and female. Option number D, all the gametes from males carry only the Y chromosome only. Means do all the sperms carry only the Y chromosome? What will be the correct answer? See what happens in XX-XY sex determination system. Males, they have one X and one Y chromosome. Females have a pair of X chromosome. So males are heterogametic. They are producing 2 kinds of gametes. Females are homogametic. They produce only 1 kind of gamete. Okay, so the option number 2 will be correct. Option number B. Males, they have 2 sex chromosomes, 1 X and one Y chromosome. That's why they are producing 2 kind of sperms. Half of the sperm will have X chromosome, half of the sperm will have Y chromosome. Now, next one, in humans, what determines the sex of the offerings during fertilization? So the gender of the child or sex of the child is determined by whom in case of humans? Okay, it is male or female, it is mother or father. So these are the options. First option is the ovum or the egg. It contributes either X or Y chromosome. Option number B, sperm contributes either X or Y chromosome. Option number C, both parents contribute equally to the sex determination. And option number D, the autosomes determine the sex. What would be the correct answer? The correct answer will be option number B. Sperm contributes either X and Y chromosome. So males, they have X and Y chromosome. Half of the sperm will have X chromosome, half of sperm will have Y chromosome. Now, the type of sperm which fertilizes or which fuses with the egg determines the sex of the child. If the sperm carrying X chromosome, this fuses with the egg, then the zygote will have XX condition and this will develop into female child. If the sperm having Y chromosome fuses with the egg, then the zygote will have XY and this will develop into male child. Clear? Now, next one, which type of sex determination is observed in birds? So, what are the options? First option is XY system with male heterogamety. Option number B, ZW system with female heterogamety, means females are producing two kinds of gametes here. Option number C, exo system with male heterogamety. Yes, this X0 system means what? X0 means one X is missing, one X chromosome is missing. Option number D is haplodiploid system. What would be the correct answer? Okay, so here the correct answer will be ZW system with female heterogamety. Okay. So here in ZZZW system, what happens? Males have ZZ chromosome, females have ZW chromosome. So females are producing two kinds of eggs here. Okay, two kinds of gametes. So this is opposite of XY sex determination system. Now next, in honeybees, the drones which are the males develop from. Options are fertilized egg and are diploid. Option number B, unfertilized egg and are haploid. Option number C, fertilized egg and are haploid. And option number D, unfertilized egg and are diploid. So what would be the correct answer? Okay, the correct answer. So you can see in honeybees what happens, haplodiploid system is observed. Here the males, they are haploid and they develop from unfertilized egg. The females are diploid because they develop after the fertilization. And the males, they have half the number of chromosomes as compared to females. Now next, X0 type of sex determination system, which of the following is true regarding males and females? So X0 type of sex determination system, this is observed in insects such as grasshopper and cockroaches. Clear? Now, what is true regarding this X0 type of sex determining system? Option number 1, males have 2X chromosome. Females have only 1X chromosome. Option number C, males have 1X chromosome and females have 2X chromosome. Option number D, both male and females have the same number of autosomes. What will be the correct answer? Option number C will be the correct answer here. You see what happens in X0 type of sex determination system. Males, they have only 1X chromosome. The 0 means they are lacking 1X chromosome. And females have 2X chromosome, or you can say a pair of X chromosome. So males, they have 1X chromosome less as compared to females. So suppose males are having 17 chromosome, females will be having 18 chromosomes. Clear? Now next, which scientists propose a chromosomal theory of inheritance? So this is a quite straightforward question. They are just asking you the name of the scientist who gave this theory of chromosomal theory of inheritance. Options are Gregor Mendel, Thomas Hunt Morgan. Option number C, Sutton and Bovary. Option number D is Alfred Sturtevant. What will be the correct answer? Option number C will be the correct answer. Sutton and Bovary, they gave chromosomal theory of inheritance. Clear? Mendel, he is known as the father of the genetics. He gave law of inheritance. Morgan, he gave the concepts of linkage and recombination by working on Drosophila. And Alpha Sturtevant, he was a student of Morgan, and he used recombination frequency to find out the distance between the genes, and he used this to create genetic maps. Now next, in Morgan's dihabit cross experiment, the deviation from Mendel's 9:3:3:1 ratio was observed due to. So you see Morgan, he was carrying out dihabit crosses in Drosophila melanogaster, and he was studying the sex-linked inheritance. Now what he observed in the F2 generation, he observed a deviation from the Mendel's typical phenotypic ratio of 9:3:3:1. So this was due to what we can say, what phenomena? First is incomplete dominance, second option is codominance, third is linkage of genes, fourth is multiple alleles. What would be the correct answer? Option number C is the correct answer here, linkage of genes. Clear? So when the genes when genes are linked, so when the genes are present on the same chromosome and when the distance between the two genes is less, then these genes are inherited together and more parental types are formed as compared to non-parental. Okay, so deviation in the F2 generation was due to linkage of the genes. The genes were not assorting independently. You get this ratio, phenotypic ratio 9:3:1, when the genes are assorting independently. But when the genes are linked, then the genes or the characters, they don't assort independently. Now, a dihabet cross is made between two parents. The genotype of one of the parent is capital R, capital R, capital Y, capital Y. So this is homozygous dominant for both the traits. Second parent is homozygous recessive for both the traits, having the genotype small r, small r, small y, small y. Now, what is the genotype of the F1 generation? So these are the four options. What would be the correct answer here? The correct answer will be option number 1. So you can see, you can make the gametes of both the parents. Okay, you can use the formula 2 raised to power n, where n is the number of heterozygous pair. So in both of these parent, number of heterozygous pair is 0. So they will produce only one type of gametes. Homozygous dominant parent will produce all the gametes having these two allele, capital R, capital Y, and the other parent will, this parent homozygous recessive, this will produce all the gametes having small r and small y. Now diploid organism, it inherits one of the allele from father, one allele from the mother. So it will inherit capital R from father, small r from mother, and then capital Y from father or and small y from the mother. So the genotype will be capital R smaller, capital Y small y, means there will be heterozygous condition in the F1 generation. The next question, which law of Mendel is illustrated by the independent segregation of two pair of genes during gamete formation? Is it law of dominance, law of segregation, law of independent assortment, or law of linkage? What would be the correct answer? Option number C, law of independent assortment, will be the correct answer. Okay, so law of independent assortment can be explained using a dihabit cross, and due to law of independent assortment or due to independent segregation of the two pair of genes, you get new combinations in the F2 generation as well. Okay, so we have discussed the mock test now. Now, let's discuss some practice questions related to the chapter principle of inheritance. Okay, clear? So let's start with these questions now. So this is the first question here. This is the first practice question. Okay, so a dihybrid cross is being made between a homozygous dominant parent with a homozygous recessive parent. The genotype is also given to you. Okay, now the ratio of genotypes, capital A small a, capital B small b, and capital A small a, capital B in the F2 generation is clear. So you have to, you can say, you have to calculate the probability, you have to calculate the ratio of getting these genotype in the F2 generation. These two genotypes have been given to you. So answer will be, okay, options are 4:2, 2:1, 1:1, and 2:4. What would be the correct answer? Okay, so let's have a look how you can solve this question. So answer is 2:1 here. Let's see how we get 2:1. Now the parental generation is given to you. These are the genotype of the parents. Then parent 1, which is homozygous dominant, having genotype capital A, capital A, capital B. This will produce only one type of gamete. All the gametes will be having capital A and small b allele. Then the second parent similarly, which is homozygous recessive, this will produce only one kind of gamete, having small a and small b allele only. F1 generation will be heterozygous, capital A small a, capital B small b. Now, after selfing of the F1 generation, okay, you will get the F2 generation. Now, how to get the F2 generation? First, you can say, you should know the gametes produced by each of the parent now. Now, each of the parent, as they are heterozygous, both of them are heterozygous, they will produce 4 kind of gametes. Again, you can calculate the gamete using formula 2 raised to power n, where n is the number of heterozygous pairs. Okay, here you have 2 heterozygous pairs. So 2 raised to power 2 will be 4. 4 kinds of gametes will be produced. Now, you can make a Punnett square now. Okay, on one side you can write gametes of male, another side you will write the gamete of females. You can now calculate all the possible genotypes. Now in the F2 generation, after analyzing this genotypes in the F2 generation, you can see capital A, small a, capital B, small b appears 4 times in this Punnett square. Okay. Now the other genotype, capital A, small a, capital B, capital B. So they were asking about these two genotypes in the F2 generation. This appears only 2 times in this, you can say these possible genotypes. So you can say the ratio is 4:2 and you can reduce this to 2:1. So 2:1 is the correct answer here. Now next one, man is having the blood group A and this man is married a woman having blood group O. A child is having blood group O. What will be the genotype of parents? So these are the 4 options given to you. A, B, C, D. What will be the correct answer? So answer will be option number B, IA small I and IB small I. So means the father is heterozygous for blood group A, mother is heterozygous for blood group B. Clear? So now they are saying child is having blood group O. Means the child will have a genotype small I, small I. Now, the child will inherit the recessive alleles from the parents, one from the father, one from the mother. So this means both the parents should have at least one I allele, one small I allele. Means the parents are heterozygous and the father is having blood group A. So he'll have the phenotype IA small I. Mother is having blood group O and she will also be heterozygous having the genotype capital I, B and small I. Okay, the parent genotype will be IA small I and the mother's genotype will be IB small I. Now you can make a Punnett square and then you can see after analyzing the Punnett square, you can see one of the child is having small I genotype. So this child will be having a blood group O. The genotype of other offerings are AB, okay, having IA, IB. Okay, one of the offerings will be having IA small I, this will be A blood group. And one of the offerings will be having IBI genotype and the blood group will be B here. Okay. Now next question. Now the two genes R and Y, they show independent assortment. Okay. So these genes, if they are showing independent assortment, they are located on, what are the options? Same chromosome, but far apart. Different chromosomes. Option number C is same chromosome close together. Option number D is different alleles. Are they two different alleles? What will be the correct answer? Yeah, the answer will be different chromosomes. So these R and Y, these two genes, they are located on different chromosomes. Then only they will show independent assortment. Okay. When the genes are present on same chromosomes, they will show linkage. Clear? So independent assortment occurs when genes are located on different chromosomes or very far apart on the same chromosomes, more than 50 map units. In this case, different chromosome is the correct answer. The next question, what is the probability of a colorblind woman being born to a carrier mother and colorblind father? So mother is carrier for color blindness and she is marrying a man who is colorblind. So father is colorblind and the mother is the carrier. So what is the probability that the daughter will be having color blindness? So the color blindness is X-linked recessive disorder. Okay. Clear? So now when the daughters will be colorblind, when the daughter will inherit both the X chromosomes having color blindness gene on them, because females, the daughters, the women, they have X and X chromosome. They have a pair of X chromosomes, and this is a recessive disorder, which are observed only in the homozygous condition. So female, the daughter should inherit both the X chromosome having color blindness gene on them. So what is the probability that daughter will be colorblind? So these are the options: 0%, 25%, 50%, or 100%. So let's have a look at the answer. Answer will be 50%. Now you can see mother is a carrier. Okay, means the genotype will be XN and XCB. N is for normally, CB is for the color blindness allele. Okay, father is colorblind, so father is having the genotype XY. Okay, XCB and Y. Clear? So the father or the male, he is colorblind. So even a single color blindness gene which is present on X chromosome is enough to exhibit this disease because there is no normal gene on the Y chromosome. Now you can make a Punnett square. Now, after analyzing this Punnett square, what you can see here. Okay, these are the 4 genotypes that you get. These are the 4 possibilities. Clear? Now you can see. Here, this is the daughter which is colorblind. She has inherited both the X chromosome having color blindness genes, one from father, one from mother. Now you can see from this Punnett square, XCB represents the colorblind daughter. XN and XCB represent the carrier daughter. XCBY represent a colorblind son and XNY represent a normal son. Now you can see why the answer is 50%. The probability of having colorblind daughter having this genotype is 1 out of 2. Okay, 1 out of 2. So there were 2 daughters. Okay, probability is 1 out of 2, which means 50%. Next question, inheritance pattern shown by hemophilia is. Hemophilia is a genetic disorder, okay, where there is a continuous bleeding because there is a, you can say, there is no synthesis or less synthesis of the clotting factors. What are the options? Y-linked, autosomal dominant, X-linked recessive, or autosomal recessive? What would be the correct answer? Okay, answer will be X-linked recessive disorder. So genes for hemophilia is located on the X chromosome. Okay. Now, there is a mutation on the X chromosome which causes hemophilia. Okay, and this mutation is recessive. That's why it is X-linked recessive disorder. So this disorder is only, you can say, okay, it is more common in males because males have only X and Y chromosome. So a single mutation on the X chromosome can, you can say, lead to this disease because there is no normal chromosome on the Y chromosome. Females are usually carriers because females have a pair of X chromosomes. So even if there is a one mutated gene on one of the X chromosome, there is a normal gene present on the other X chromosome. So females are usually carriers. Here you can see image showing you hemophilia. In the normal case, what happens? The blood clots after some time, but in case of hemophilia, the blood is not able to clot because the clotting factors are not being produced. Okay, or the blood, or it will take a very long time for blood to clot. The next question, F1 progeny is being crossed with the homozygous recessive parent. So this type of cross is called a test cross, back cross, monohybrid cross, or both A and B. Means both test cross and back cross. So what will be the correct answer? Let's have a look at the answer. See, answer here will be both A and B. Let's have a look at the explanation. So see, test cross is a cross between F1 hybrid and the homozygous recessive parent. Now, what is back cross? Back cross is a cross between F1 hybrid and any of the parents. So in back cross, the F1 generation is crossed with any of the parent. Clear? Now, why answer is both A and B? Because if F1 is crossed with homozygous recessive, so when Mendel did the experiment, he used the parents. Okay, two parents. One of the parent was homozygous dominant, another parent was homozygous recessive. So in back cross, you are crossing F1 hybrid with any of the parents. So you can even cross with homozygous recessive parent also. So back cross comes under, you can say test cross come under back cross only. Clear? In back cross, we can cross F1 with homozygous dominant also and with homozygous recessive also. If the parent is homozygous recessive, we call it a test cross. We have given it another term. So answer can be both A and B. Clear? When the parent is homozygous recessive, it can be both test cross and back cross as well. If there was no option of back cross here, then the answer will be test cross. Now, this is a very interesting question. Humans' ability to roll tongue is dominant, controlled by capital R, over non-rolling capital, controlled by small r allele. Now, in a family, father can roll his tongue, but mother cannot. Now, among their 4 children, only 2 can roll their tongue. The genotype of the father is. These are the options given to you. Option number A is capital R, capital R. Option number B is capital R, small r. Option number C is small r, small r. Option number D is capital R, or small r, capital R. What will be the correct answer? Let's have a look at the answer. See, answer will be option number B, capital R, small r. Let's have a look at the explanation. See, mother's genotype is small r, small r. Okay, because mother cannot roll his tongue. Okay, so if in the question it was given mother cannot roll the tongue. So mother will be homozygous recessive. Now father was capital R, capital R. Okay, because father can roll his tongue. Clear? And in the question it is given the ability to roll tongue is dominant over non-rolling. So if the father was capital R, all the children will be having the genotypes capital R, small r means all will be rollers. All can roll the tongue. But they are saying among 4 only 2 can roll the tongue. So this means the father should be having capital R and small r genotype, not capital R, capital R. Yes, since only 50% of children can roll, father must be capital R and small r. So you can make a Punnett square. So this is the genotype, this is the gametes produced by the female. Okay, because female is capital R, capital R. Male, it will produce 2 kind of gamete. Half of the gamete will have capital R, half of the gamete will have small r. Now you can make a Punnett square, and after analyzing Punnett square, you can see here 2 can roll their tongue only. Clear? So this you get capital R, small r, capital R, small r. 2 of them can roll the tongue, 2 of them cannot roll the tongue. So 50% of the children can roll the tongue, 50% of children cannot roll the tongue. And this is why, because the male will be capital R and small r. Next question, chromosomal theory of inheritance was proposed by option Mendel, Morgan, Sutton, Bovary, Watson and Crick. This is a very straightforward question. What is the correct answer? So let's have a look at the answer. Answer will be Sutton and Bovary. So let's have a look at the explanation. So Sutton and Bovary independently proposed chromosomal theory of inheritance. What they did, they established, okay, a parallel relationship between chromosome behavior and Mendelian factors. Okay. So what they said, the genes are present on the chromosomes and the behavior of chromosomes and genes, they are parallel to each other. Okay. So they explained Mendelian's inheritance laws with the help of chromosome movement. So they established a parallel between chromosome behavior and Mendelian factors. Mendelian factors are nothing but genes. That both chromosome and genes, they behave in a similar manner because genes are present on the chromosomes. They demonstrated chromosomes are carriers of the genetic material, means genes are present on the chromosomes. So here you can see this is a chromosome movement during meiosis. Okay, this is showing you the cell cycle here. This is showing you meiosis. First is the G1 phase, then you have the G2 phase. The chromosome has doubled now. In the anaphase of meiosis 1, chromosomes separate and the two cells are formed. In anaphase 2 of meiosis 2, the chromatids separate and in the end you get 4 cells. Okay, so this is chromosome movement during meiosis. Clear? So by this chromosome movement, Sutton and Boveri said, or they explained the Mendel's laws of inheritance using this chromosome movement. Now, next question, a marriage between a normal man and a carrier woman for color blindness will result in. So here you can see the father is normal. Woman is a carrier. So this will result in all normal children, all sons colorblind, 50% son colorblind, and 50% daughters colorblind. What will be the correct answer? So let's have a look at the answer. Option number C, 50% sons will be colorblind. This is the correct answer. So you can make a Punnett square here. Okay. One side you can write the gametes produced by female, another side you can write gametes produced by male. So here you can see this capital C is for normal and this C, which is highlighted with yellow, this is showing you the color blindness gene. So female is a carrier. So one of the X chromosome will carry normal gene, another X chromosome will carry color blindness gene. Male is normal here, father is normal. Okay, now you can make different combinations. You can see here the sons. Let's look at the genotype of the sons. One of the son is normal, another son is colorblind. So that's why the answer is 50% sons will be colorblind. 1 out of 2 sons are colorblind. Okay, so 50% of sons get the colorblindness. The next question, a 28-year-old pregnant woman undergoes amniocentesis. So with the help of amniocentesis, what you can do with amniocentesis? You can detect the genetic disorders. Yeah, and the karyotype analysis revealed that her fetus has 47 chromosomes instead of 46 chromosomes. And there is extra copy of chromosome 21. Which of the following condition in the fetus is most likely to have? Okay, so options are Turner syndrome, Klinefelter syndrome, Down syndrome, or Edwards syndrome. So you can see there is a trisomy of 21 chromosome. There are 3 pair of 21 chromosome. There is extra copy of 21 chromosome. Usually there are 2 copies, but there is 1 extra copy of 21 chromosome, means there is 3 copies. This is known as trisomy of 21st chromosome. What will be the correct answer? Okay, here the correct answer will be option number C, Down syndrome. Okay, so Down syndrome, known as trisomy of 21 chromosome, is characterized by the presence of an extra copy of autosomes, extra copy of chromosome 21, resulting in 47 chromosome instead of normal 46. So here you can see these are the symptoms of a child having Down syndrome, and the karyotype is also shown to you. Here you can see there is a 3 copies of the 21st chromosome. Okay, clear? So with this, we end this session. I hope this session was quite knowledgeable for you. We'll meet you in the next session. Thank you very much for listening.